When our biology student first learned she had red-green color vision deficiency, she wanted to understand how she got this trait. Why could her friends see colors that she couldn't? But there's a story that's even bigger than the inheritance of a single trait. It's the story of how chromosomes are the basis of inheritance. Chromosomes are cell structures that carry genes from parent to offspring, and their behavior during meiosis and fertilization is what generates the incredible variation we see in every generation and determines a multitude of traits including blood type, hair color, skin tone, and whether or not you can see all the colors of a rainbow. Hi everyone, I'm Noel Poller, AP Biology teacher and author of the Lab Hamster Storylines. As we near the end of Unit 5 exploring heredity and genetics, we're now going to connect chromosomes to inheritance and explore how segregation, independent assortment, and fertilization generate genetic variation in populations. We'll also look at how errors in the system can lead to genetic disorders. Let's start with segregation. During meiosis, homologous chromosomes are separated into different gametes. This means that each gamete receives only one chromosome from a homologous pair, and therefore only one allele for each gene. That's why Mendel's law of segregation works. It's rooted in the physical behavior of chromosomes during meiosis 1. This relates to color vision deficiency because the sex chromosomes carry the genes associated with red-green color blindness. To be specific, the genes for red-green color vision deficiency are located on the X chromosome, but these genes are missing from the Y chromosome. Next comes independent assortment. During metaphase 1, homologous pairs of chromosomes align randomly at the metaphase plate. Each pair sorts independently of the others, meaning maternal and paternal chromosomes are mixed in countless ways. For humans with 23 pairs of chromosomes, the possible combinations of chromosomes in gametes number in the millions. And that's before even considering the impacts of crossing over, which adds even more variation. This is important for the inheritance of color blindness because it means that an egg cell will end up with only one X chromosome, and a sperm cell will have either one X or one Y chromosome. The third source of genetic variation is fertilization. Each parent contributes one gamete. The zygote receives a combination of chromosomes from two different individuals. The probability of two zygotes with identical chromosome combinations forming is astronomically small. Fertilization ensures that every zygote has a one-of-a-kind genetic makeup. To be colorblind, a female must inherit two copies of the recessive allele for red-green color vision deficiency, one on the X chromosome in the egg, one on the X chromosome from the sperm. Together, segregation, independent assortment, and fertilization explain why siblings are genetically unique, even when they share the same parents. A female with red-green color vision deficiency can be traced back to the segregation and inheritance of two X chromosomes carrying the recessive photopigment gene. Her brother inherited one X chromosome with the non-mutated photopigment gene, and because the photopigment genes are not located on the Y chromosome, he has typical color vision. But chromosomes don't always behave perfectly. Sometimes mistakes occur, leading to genetic disorders. One type of mistake is called non-disjunction. This occurs when chromosomes fail to separate properly during meiosis. If this happens in meiosis 1 or meiosis 2, gametes can end up with too many or too few chromosomes. When such gametes are involved in fertilization, the resulting zygote may have chromosomal disorders. For example, trisomy 21, Down syndrome, occurs when an individual inherits 3 copies of chromosome 21. Other disorders like Turner syndrome or Klinefelter syndrome result from non-disjunction of sex chromosomes. Some human genetic conditions, however, come from inheriting a single mutated allele rather than a whole chromosomal change. For instance, sickle cell disease results from a single base change in the hemoglobin gene. Color vision deficiencies also fall into this category, where a single gene mutation on the X chromosome alters the ability to distinguish between certain colors of light. So the chromosomal basis of inheritance provides the framework for understanding why traits are transmitted the way they are, why each individual is unique, and why sometimes errors in chromosome behavior can lead to genetic disorders. Thanks for watching. I hope this helped you see how chromosomes drive genetic variation in sexual reproduction and explain both the diversity and the challenges that come with inheritance. And what about the biology student? Was she colorblind due to a mysterious environmental factor? Was her dad right when he thought that he wasn't actually her biological father? I hope you'll check out my final video with Paul Anderson to find out. It's right here.